Restriction enzyme cloning is the one of the earliest and relatively straightforward cloning techniques. It allows to cut both the vector and insert at specific DNA sequence that glued them together to create recombinant DNA molecule. This technique depends on restriction enzymes. In 1960, scientists discovered that certain bacteria could defend themselves against invading viruses by restricting or cutting foreign DNA, such as bacteria-phage DNA. Soon after, in the early 70s, the first restriction enzyme was isolated. The enzyme recognized specific DNA sequences and cut DNA in a highly precise manner, producing either sticky ends or blunt ends. This precision is what made restriction enzyme an essential tool in recombination DNA technology. Now let's walk through how to plan a successful restriction enzyme cloning experiment using a simple example such as cloning the EGFP gene into PUC19. There are three main steps. First, prepare your vector. Second, prepare your insert, and third, bring them together using a ligation reaction. Start by deciding where you want to insert your gene, then identify restriction enzymes that will cut the linearize your vector. This can easily be done using software like SnapGene. Let's have a look. Open your vector sequence, locate the multiple cloning site, which is bunch of restriction sites next to each other designed to facilitate cloning, and select enzymes that cut in this region. I am going to select S3 echo R1. Make sure each enzyme cuts only once, otherwise your plasmid will be fragmented, and you can do that by displaying on only the enzymes that cut once. So under enzymes, make sure the unique cutters is what is selected. Once you have chosen your enzymes, keep three important tips in mind. First, select enzymes that are available in your lab to save yourself some time. Second, make sure both enzymes work in the same buffer so you can run a single digestion reaction. And third, confirm that this enzyme do not cut within your insert sequence. So just quickly open your insert sequence. Again, enzyme that are unique cutters, go to map and make sure that enzymes you have selected are not actually present within the insert sequence. Now that you have selected your digested enzyme, set up your digestion according to manufacturer's instruction, and after digestion, run the sample on the agarose gel and gel purify the linearized vector. Uncut plasmid remain supercoiled and migrate differently on the gel. So purification ensure you isolate only the correct cut vector. Next, we prepare the insert fragment. The goal is to add sticky ends that match those on the plasmid, which can done through careful primer design and PCR amplification. Let's draw this out. Starting with your gene of interest, you can either order the open reading frame directly or amplify it from cDNA. First, we'll add restriction size to both ends of the gene. On the 5' end, S3 recognition. On the 3' end, the echo R1 recognition sequence. Upstream of each restriction site, we'll also include a short 4 to 6 base pair leader sequence. These extra bases are random, but they are important because restriction enzyme cut more efficiently when the recognition site is located within the DNA strand rather than right at very end. Once your map is out, SnapGene designing and ordering primers become much easier. Open the GFP sequence on SnapGene. Let's start by adding the S3 recognition sequence first. Place your cursor at the start of the gene, then go to edit, insert, restriction site, and search hint 3. Insert to be organized. Let's add it as feature. Select it and go to the feature, add feature, label it restriction site. Now let's add echo R1 recognition sequence. Go to the end of the gene on the and where it is present, place your cursor at the end and again edit, insert restriction site and search for echo R1. Highlight, add feature, restriction site. Next is to add the leader sequence. Place your cursor right at the end, go edit, insert bases, type out 4 to 6 random bases, select insert. I like to label my all my features. And now we'll add one at the end also. Edit, insert bases, paste, insert, feature, add feature, leader sequence. Now that we have mapped everything out, let's design our primers. First, choose the gene specific complementary sequences for your forward and reverse primers. try to keep their melting temperature similar, ideally within about 2 to 5 degrees Celsius of each other. Let's do that, highlighting them. So if I select around 20 base pairs at the start, I can see the melting temperature is between 61 and 64. Let me check at the end of the gene, that's around 53 and 54. So there is quite a big difference. So I am going to play around with the primers to ensure that the difference in degrees is within the range that I had mentioned earlier, 2 to 5 degree. So if I add a few over the here, I can bring it up to around 60 degrees. So that's 23 base pair. And then let me see over here, if I just bring it down to 18 base pairs, that's around 60 degree. I'm just going to highlight this so that I can keep track of them. Select the base pairs that I want. I am going to add the primers and I'm just going to label this F1. So this is not the primer that's going to be ordered, but I am just labeling so that I can keep the track of the bases that I want within my primer design. And then I mentioned over here that we are going to ahead and select around 23 base pair, so that is around 60 degree as well. I'm going to go to primers and primer bottom stand R1 again. Just know that this is not the primer that's going to be ordered, but I just would like to keep track of the so that I know the bases which I have selected. Now that I've highlighted the sequence that is complementary to the gene for the forward and reverse, I'm going to ahead and select all of these, including the restriction sites and leader sequence and primers. So this is would be the primer that I will order, that will integrate the restriction site and leader sequence during PCR amplifications. So I'm going to call this forward primer, and then I will do the same at the bottom primer. Add primers bottom stand reverse primer. Now if you go to the primers, you can find the primer sequence for the primer that you have designed. So here's the forward primer. You can just go ahead and copy this and paste it into your order form as well. Do the same for the reverse primer. Now that you have designed your primers and ordered them, you are going to ready to set up your PCR reaction. Just note, when you come to set the annealing temperature, use the melting temperature of the sequences that are complementary to the gene. That is important, not the additional sequence that will be incorporated during amplifications. After a successful PCR reaction, digest the PCR product with the same restriction enzyme to generate matching stickings. Then purify the digested insert using a DNA cleanup kit or gel purification step. If you have the paid version of SnapGene, you can actually simulate restriction enzyme cloning to see what at the end product you will get. Let me walk you through the open your vector file. Go to the action, restriction and insert, cloning insert fragment. Over here, type in restriction enzyme that we are using in our example. So hind 3 and echo R1. Make sure the orientation that's selected is correct. Next, go to the fragment on the top over here and choose the file. So make sure that you choose the file where you have added the restriction recognition site as well as the leader sequence. Cut with the same restriction enzymes. So hint 3 and echo R1. Select clone. This is what the final product should look like. If everything goes well, where you have your GFP gene under the control of the lag promoter, this example and the size of the product is around 3 kb, 3.3 kb. Finally, mix your digest vector insert with DNA ligase, recommended using T4 DNA ligase, and following the manufacturer's protocol. I'll include the link below. The ligase conveniently joins the compatible ends to form a recombinant plasmid, which can then be transformed into bacteria to get the colonies.